Beadle and Tatum's work pretty much established that genes code for proteins. That is, the idea is that information in the sequence of nucleotides in a DNA molecule is somehow used to specify information, a sequence of amino acids in a protein molecule. Now you should already know that this works by two steps. First one is transcription, which takes information in the DNA and uses it to synthesize an RNA molecule. And then translation takes information in the RNA molecule, in a messenger RNA, and uses that information to specify the sequence of amino acids in a protein. We're going to start out then by looking at the process of transcription in some detail. Man, I really need to check my hair before I record these things. The other one, I was having a bad hair day yesterday. So let's think about the process of transcription in the bacterium E. coli. Here you've got the E. coli molecule. This is the DNA molecule of E. coli, the whole genome, which is 4.5 million base pairs. As you know, it has about 4,400 genes in there. And imagine that you are RNA polymerase, the enzyme that's going to synthesize the RNA. Here you are, you're all set, you're going to come along here, and what's your first problem? Well, your first problem is, where do you start? You've got 4,400 genes in there. You're going to transcribe one of them. Where is that gene? Where's the beginning of a gene, the starting point for it? And the answer to this question is that each gene has a sequence in the DNA that's a binding site for RNA polymerase. This is a place where RNA polymerase can bind and start transcription. And this special sequence that signifies where you start a gene is called the promoter. Okay, so here we have the E. coli genome, and now I've marked on it some genes. And of course, I haven't marked as many as there really are. There's 4,400 of them, but I've given you the idea here. And if we enlarge one of those and take a look at it, here's a typical gene then. The green part, that's what we call the coding region of the gene. This is the part that codes for protein. It'll be transcribed into a messenger RNA, and that'll be translated to give you the protein. The little yellow part, and it's not really to scale, it should be a lot smaller, but that little sort of butterscotch colored part at the beginning, that's the promoter. This is a sequence that doesn't code for protein. It's not even transcribed. All it is a place in the DNA where RNA polymerase is going to bind to get started in the process of transcription. In E. coli, this promoter sequence has a particular characteristic sequence. So, what's the DNA sequence in E. coli where RNA polymerase binds? What's the E. coli promoter? Those of you who had me in a class long ago and far away may remember that this is TT Gaka Tata cat. Well, and that's pretty close to true. I only lied to you a little bit about that. If you look at the place where transcription starts, and that would be the boundary here between the yellow region and the green part, which is going to be the coding region of the gene. Okay? The start point of transcription, then, the very first nucleotides transcribed, when I showed you this previously, you saw that as CAT. Actually, it's more general than that. It's pyrimidine, purine, pyrimidine, purine, pyrimidine. Okay, YRY. By the way, Y is the one-letter abbreviation for pyrimidine. R is the standard one-letter abbreviation for purine. So YRY. And you can see that CAT, C-A-T, is an example of YRY. The first nucleotide transcribed, then, is that R. It would have been the A in CAT if you had CAT there, but whatever that R is, that's the first nucleotide that actually gets transcribed. The rest of, so we then can talk about the gene, the portion that is transcribed going to the right from the start point there. We call that direction downstream. The part that is before that, going the other direction, we call that upstream. And the upstream region is where most of the promoter is. Only two nucleotides of the promoter, only that R and Y, actually get transcribed. All the rest of it isn't transcribed, but that's where RNA polymerase binds to start. Now, we talk about positions in a gene. If you want to talk about a particular nucleotide somewhere in a gene, this one right here, well, maybe it's the 30th one counting from the starting point. That would be plus 30, or number 30 is that nucleotide. The first one transcribed, that R, that one's plus 1. The Y before that R, that one is minus 1. And if you go further back, we give those negative numbers going upstream. So as you look back from there, around the minus 10 region, somewhere in that vicinity, you see this sequence TATA-AT, T-A-T-A-A-T. If you go further back to the minus 35 region, you find TT-GACA, T-T-G-A-C-A. These are the consensus sequences found in the E. coli promoter, TT-GACA, TATA-AT, YRY. They have to be, the nucleotides in between, where you see the dots there, can be anything. The spacing's important. In other words, number minus 35 has to fall somewhere in that TT-GACA. Minus 10 has to fall somewhere in the TATA-AT. But what the nucleotides are in between those chunks, they can be anything. Okay. So RNA polymerase is going to bind there and start transcription going to the right. So the promoter actually specifies for RNA polymerase where transcription is going to begin and which way it's going to go. Garon, those extra two nucleotides, no, they don't code for protein. In fact, the beginning of a gene, if you look at, here, let me draw on it. Okay, so at the beginning of the gene here, or I'm sorry, the beginning of the transcript is actually before the beginning of the part that codes for proteins. So there's always an untranslated region at the beginning of the transcript. We refer to this as the 5' untranslated region. And so those two nucleotides aren't really doing much of anything in the RNA. They're really just there as part of the whole sequence that gets transcription started. Okay, so here's the consensus E. coli promoter. By the way, this slide here, please look at it in the PowerPoint. Print out the whole PowerPoint and look at it. The subscripts didn't come out very well in this version, so it's kind of confusing looking. I'll sort of lead you through it. So the minus 35, remember, is TT-GACA. T-T-G-A-C-A. The numbers next to those numbers indicate what percentage of promoters would have that base in that spot. So for instance, over here where you see T69, 69% of E. coli promoters, and this has been determined by looking at them all, 69% of E. coli promoters have a T at that position right there in the TT-GACA. There can be some variation, right? 79% have a T in the next spot, and so on. If you look at the minus 10 region, also sometimes called the Pribnow box after the guy who discovered it, that's T-A-T-A-A-T. And again, you see the percentages of promoters that have those specific nucleotides in those specific spots. Now, it's maybe a little misleading to think about this. Although none of those nucleotides has to be the same, you can change any one of those nucleotides and it'll still be a promoter that'll function. And it may function better the closer it is to this consensus sequence. You can't change them all. You don't have a promoter if you happen to have none of those bases there. So not that many promoters will differ by more than 2 or 3 nucleotides from the consensus promoter. However, different ones would have different ones that vary. Finally, down here at the bottom, of course, you have YRY. Minus 1 is Y. 1 is R, 2 is Y. So pyrimidine, purine, pyrimidine, right around the start point of transcription.